In this video, we're going to talk a little bit about how do cells regulate which genes get expressed and which ones don't. Because as we know, specialized cells only have to produce certain proteins in order to do their job, not all the proteins that living thing can make. So let's talk about prokaryotes first. So prokaryotes typically work through operons, a group of genes that are regulated together. So these genes have a related function, and they're controlled by a single promoter. So DNA binding proteins in prokaryotes regulate genes by controlling when transcription occurs and when it doesn't. One of the keys to transcription bacteria is the organization of genes into these operons. An example of this, the lact operon. When lactose is not present, the cell doesn't need to produce the enzymes that break down and digest lactose, so the lact genes are turned off by a regulatory protein called a repressor that binds to the DNA and blocks transcription by blocking RNA polymerase. So here's kind of how it's organized. You've got a repressor gene that codes for the protein, the repressor protein, which is active because it's what we call an inducible operon. It's normally off, but can be turned on. The promoter is where RNA polymerase would normally bind. The operator is where the repressor binds when it's active. And then you have the structural genes, in this case, the lactose. So there are two regulatory regions. There's the promoter, which when RNA polymerase can bind, allows the enzyme to be produced, and the operator, which is where the repressor binds and blocks RNA polymerase and shuts off the promoter. When the lac repressor binds, like I said, RNA polymerase cannot reach the genes and cannot do transcription. When lactose is present, it binds to the repressor, changing the shape of the repressor so now it no longer fits with the operator. RNA polymerase can bind, transcription, translation, enzymes for breaking down lactose are produced, and the lactose is broken down, and then once the lactose is used up, the repressor becomes active again and it shuts off transcription and translation of the lac operon. Eukaryotes, a little bit more complicated. You've got other things. Typically there's an area upstream in front of the gene called the Tata box, which helps position the RNA polymerase. There's also transcription factors that bind to the enhancer regions, which also work to position the RNA polymerase so that it can transcribe the gene. So by binding DNA sequences in the regulatory region of the eukaryotic gene, transcription factors control the expression of those genes. So in eukaryotes, because of differentiation and specialization, you have this very complex set of gene regulations that helps, in some cases, turn some genes off permanently in some cells, but in others, it allows genes to be on or off depending on the needs of that particular cell. So it's also important with development. Certain genes are turned on and off during different parts of development in multicellular organisms. Going from a fertilized egg to an adult involves a lot of gene regulation. So there are these related things that deal with development. There are homeotic genes which regulate organ development. Homeobox genes which code for transcription factors that activate other genes important in development. So they don't actually directly control development, but they control the genes that do. And Hox genes which determine the identity of each body section by location. Where a cell is in relationship to the body of the embryo determines which genes are turned on and off and which proteins are produced to produce those particular parts of that organism. There's also some what we call epigenetic control, which is indirectly regulated. It has to do with DNA structure. DNA that's tightly coiled around histones are turned off due to the fact that transcription factors are blocked. You also have methylation, which can can block transcription and things like that as well. So epigenetic mechanisms control which genes are on and off during each stage of an insect's life cycle as it goes from a caterpillar to the pupal stage to the adult stage. The environment can also influence which genes are regulated, things like temperature. It can influence how and when epigenetic marks are added to chromatin. So in the Himalayan bunny here, in the areas of the body where the temperature is below 25 degrees, it produces the black pigments in the ears and the nose and the tail and so on in the feet. But if the temperature gets raised and those areas don't get cold enough, it doesn't produce those colors.